at the structures of the brainstem and the ventricles. And so we're looking at the brain model here. Again, just one half of it. We're identifying here is the stalk of neural tissue that is going to technically connect the cerebrum to the spinal cord is our brainstem. The brainstem is made of three parts: the midbrain, the pons, and the medulla oblongata. Here would be the midbrain, which is the most superior part of the brainstem. This area here is the pons, and then just inferior to the pons is the medulla oblongata, and the inferior most part of the medulla oblongata is going to be continuous with the spinal cord. Two structures for you to identify that are associated with the midbrain: the superior colliculus and the inferior colliculus are both kind of posterior bumps on the dorsal surface of the midbrain. So here we see the superior colliculus and the inferior colliculus. The cerebellum, again, is this structure here. It sits posterior. It's got a posterior relationship with respect to the cerebrum. So we've got the cerebellum here. If I bring the other part of the model back into play, this is the cerebellum here, and this is the brainstem. The arborvitae, so the arborvitae, we take a look at the inside of the model. The arborvitae is this white part of the cerebellum. It almost looks like the trunk and the branches of a tree. Arbor vitae means tree of life. The vermis is the tissue that connects the two cerebellar hemispheres. So this is the vermis here. And then finally the folia are the ridges of the cerebellum. So we see the folia here. The ridges of the cerebellum, where these elevated pieces of cerebellar tissue are kind of analogous to the gyri of the cerebrum. So we've got the folia or the ridges here. You can see the folia, the internal view of the model as well. That would be the darkened area just outside of the arbor vitae. Let's now look at ventricles. So the ventricles are spaces within brain tissue that produces the cerebrospinal fluid, and through which cerebrospinal fluid travels to eventually make its way out and into the subarachnoid space. Got a model here that shows the ventricular system. So this model is showing a system of spaces that are found within brain tissue. So if we were to open up the brain and view some internal cavities or spaces within this mass of brain tissue, what we would see are these spaces. So you can think of the ventricular system of the brain. If we were to take the brain and insert a needle through the outer part of the cerebrum and inject a casting material into these ventricular spaces, like a plaster of Paris, and allow it to harden, and then open up, pull apart the two hemispheres, what we would have is a representation of the ventricular system. What we can see here, these are large, what are known as lateral ventricles. So each of these lateral ventricles sits within a cerebral hemisphere. And if we take a kind of a side look at the lateral ventricles, you can see that they're very large. So this would be one lateral ventricle. This would be the other lateral ventricle here. The third ventricle sits just between the two lateral ventricles. So here we see the third ventricle. The cerebral aqueduct is going to connect the third ventricle to this down here is the fourth ventricle. And there's a little passageway known as the cerebral aqueduct. If we take a superior look at this, lateral ventricles, the third ventricle, right in the middle, this would be right in the midline in between the two cerebral hemispheres. This is our cerebral aqueduct, and this is the fourth ventricle. Viewing the brain model, the only ventricles or parts of the ventricular system we can really make out on this brain model is this is representing the cerebral aqueduct. So again, this cerebral aqueduct here is represented by this opening, and this is the fourth ventricle, which in the ventricular model, the fourth ventricle is down here.